Hello, grade 11 students. In this video, we're going to talk about our new lesson about transcription and translation. So chromosomes at metaphase, as we said before, they are made up of two chromatids. The substance that make the chromosomes is called chromatin. Chromatin is composed of DNA and proteins. DNA composed of the nucleotide A, T, C, G. Every one of these is called a nucleotide. Every three together, they are working together. Every three, they are called triplet, or we gave them the name codon. And we said that the nucleotide A and T, they are complementary, while C and G also they are complementary. So these are the nucleotide. So every one is called a nucleotide. Every three together, they are called codon, or they are called triplet. A and T, they are complementary. C and G, they are complementary. Also, we know that the DNA is deoxyribose nucleic acid. It's double strand. So in the first strand, if you have A in the first strand, so in the second strand, you should have the complementary of A, which is T. If you have C, the complementary is G. If you have G, the complementary is C, and so on. Proteins, they are composed of histones and non-histone, but mostly my work is going to be about the nucleotide. DNA, by the process of transcription, they give mRNA. And mRNA, by the process of translation, they give polypeptide, or they give amino acid sequence. Transcription. What is transcription? Transcription is the process where mRNA is produced based on the sequence of the DNA. So based on the DNA, we will give the sequence of mRNA by the process of transcription. The produced mRNA, they undergo mutation. This mutation is our point, our lesson for the next week. We're going to talk about mutation in specific, what the meaning of mutation, what are the different types, and what do they do inside the body. So DNA gives mRNA by the process of transcription by the action of two enzymes, helicase and RNA polymerase. Consider the sequence of DNA shown below. This is the sequence of the DNA shown below. This sequence is containing the GCA, TGC, ATC, TGC, CAT, TGC, and so on. So here we have the sequence. So this sequence is called the non-template. It's called the non-template. Okay? We said that it's called the non-template. Also, it's called the non-transcribed. Okay, it's called the non-template. Also, it's called the non-transcribed, while it's called the coding. Okay, so it's called non-template. Non-transcribed is called the coding. So the sequence of this is the same as the sequence of mRNA. Instead of T is replaced by U. Why? If we want to write complementary strand, the complementary of G, it's C. The complementary of C, it's G. The complementary of A, it's T. And the complementary of T, it's A. The complementary of G, it's C. The complementary of C, it's G. Let's continue. A, the complementary, it's T. The complementary of T, it's A. And the complementary of C, is G. Also, let's continue. Complementary of T, it's A. And the complementary of G, it's C. And C, complementary is G. Let's continue. C, the complementary is G. A, complementary is T. T, the complementary is A. And we have here the last codon. T, the complementary is A. And G, the complementary is C. And C, the complementary is G. This sequence is called the opposite of the first. The first one was called the non-template, while here is called the template. Non-transcribed here is called the transcribed, and it's the non-coding sequence. So you see here, every three they are called codon. Okay? Every three they are called codon. For example, if I said the third codon, where is the third codon? This is the first, this is the second codon, and here the third codon. Fifth codon, this is the fifth codon. Sixth codon, this is the sixth codon. Fourth and second codon. Okay, so here you have to be careful about the names. So non-template is called the non-transcribed, it's the coding. First part, they said write down the sequence of the mRNA. sequence of the mRNA. We said that it's the same as the coding, and instead of T, it's replaced by U. So it's going to be GCA. Instead of T, UGC, ATC, AUC, TGC is going to be UGC, CAT, CAU, TGC, UGC. Okay, so here, write down the sequence of the mRNA. This is the sequence, but you have to know that the non-template strand is called the non-transcribed and is called the coding, while the template strand is called the transcribed and it's the non-coding. In order to write the template, you have to make complementary as we did here. So this is the sequence. If you want to write the sequence, so this is the sequence of the template. Here we have first CGA, CGT, ACG, TAG, ACG, GTA, and ACG. Non-template and the template. mRNA sequence is similar to that of the coding. It's similar to that of the template. Instead of T, it's replaced by U. Instead of T is replaced by U. Instead of T, replaced by U. Instead of T is replaced by U, and so on. So here we have to know that the mRNA strand is complementary to the template, and we have to know also the mRNA is identical to the non-template, but T is replaced by U. So here we have the non-template, the sequence complementary GCA, the complementary is GC, CG, AT, TA, CG, and GC, AT, TA, CG, and so on. The mRNA sequence is similar to the non-template, which is called the non-transcribed and it's called the coding, but T is replaced by U. So GCA, GCA, TGC, it's UGC, ATC, AUC, TGC, UGC, T is replaced by U, CAT, CAU, T is replaced by U, TGC, it's UGC, T is replaced by U. But wait, if I gave you the template, can I give the sequence of mRNA? Yes, sure. Template is called the transcribed, it's the non-coding. So what I have to do is to make the complementary, but instead of putting T, we put U. So C, the complementary is G. G, C. T, it's A. A, the complementary, it's T. But here we put U in the mRNA. So in the mRNA, what's the difference between the DNA and mRNA? First of all, DNA is double-stranded, mRNA is single-stranded. Second, DNA contains T, A, C, G, T, while the mRNA we have A, C, G, instead of T we have U. This you can determine that this is the sequence of mRNA. So mRNA strand, it's always complementary to the template, while mRNA strand is the same as the non-template, while the T is replaced by U. Helicase, what does it do? It unwinds the DNA of the gene coding for the protein. RNA polymerase synthesizes the complementary strand. It synthesizes or produces the complementary strand. Translation. So first process, we know that it's called transcription. Transcription is the first step of the protein synthesis, where we move from DNA into mRNA by the process of transcription by the help of two main enzymes, helicase and RNA polymerase. Translation is the second step of the protein synthesis, where an RNA produced in the cytoplasm will code for the production of proteins. These proteins, either they remain in the cell or they move out of the cell. So consider the sequence here of the non-transcribed strand. Here we have AAA, CCC, GGG, CAA, TGC. Give the sequence of the transcribed. I gave you here the coding. Coding is the non-transcribed, it's the non-template, it's the coding. While I want the transcribed, simply I can make it complementary. A, T, A, T, A, T, C, G, C, G, C, G, 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 C, G, C, G, C, C, G, A, T, A, T, T, A, C, G, G, C. This is the sequence of the transcribed strand. Specify the sequence of the mRNA coding. I need the sequence of the mRNA. I can use either the coding or the transcribed. Coding is the same. Non-transcribed, non-template is the coding. It's the same sequence as mRNA, but the T is replaced by U. Here, the same sequence as the coding, but the T here is replaced by U. So mRNA is the same sequence as the coding strand, while the T is replaced by U. Sequence of amino acid. How can I determine the sequence of amino acid? Sure, not from my own mind. I have to use the genetic code table. Do we have, Mr. Muhammad, to study these genetic code table? No, you don't have to, but you have to study only three. You have to study only three. They are called the stop codons. Okay, we said that every three they are called codon. So here they are called the stop codon. UAA, UAG, and UGA, they are called stop codons. And other, they are amino acids. You see here, for more than one codon, they code for the same amino acid. For example, leucine, proline, threonine, methionine, isoleucine, valine, alanine, asparagine, glutamine, glutamic acid, and so on. So we have a lot of them, many. Histidine, for example, we have many amino acids. They are coded by more than one codon. So let's see here first. We have AAA. Where is AAA? First letter is A. We start here. Here is the first letter. Here is the second letter, and here is the third letter. So we move like, for example, a puzzle here. First, second, third. AAA. First letter is A. Second letter is A. It's LYS. I see. So it's here. Second one, it's C. 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 So C the first letter. C the second letter is proline. PRO. Third one, it's GGG. Lysine. GGG. First letter G. Second letter is G. So here is GLY. Number 4, it's CAA. CAA. Where's CAA? This is C, the first letter. A is the second letter. CAA, GLN. And we have last one, UCG. The first letter is U. Second letter is C. UCG is serine, SER. From here, we can determine the sequence of the amino acid. You tell me that do we have to write them as a capital letter? The first letter is capitalized. It's okay here. For example, the first letter only is capitalized. You have to put between them dashes, since the first one for the first codon, second for the second, third for the third. Now, for example, if we have a stop codon, what do? Simply, if you reach, for example, let's say here, instead of UCG, we have the stop codon UGA. So you reach here the stop, you write here stop. Simply, you write here stop, and here we stop. We don't continue, even though it comes in the middle. You write stop, and you don't continue the string. Why? So the stop codon, it stops the process of the translation. There is no more amino acids in this sequence. So you have to stop at this stop codon. That's why it's called the stop codon. Okay? So you have to be careful about this. I will repeat. The non-transcribed strand is called the non-template, and it's the coding. You have to study this, and you have to keep this in your mind. The non-transcribed, the non-template is called the coding. The same sequence as the mRNA, but T is replaced by U. So here I have to know what is given in the question. Here they gave you the question and the coding sequence, okay, which is non-transcribed, the non-template, and the coding DNA strand. So here you have to know that the coding is the same as mRNA, but T is replaced by U. If they gave you the non-coding, you have to make it complementary. Instead of T is replaced by U to give the sequence of mRNA. You can use whatever you want, but you have to pay attention to the question. The question can solve most of the exercise if you pay attention. If you know that non-transcribed, the non-template is called the coding, and the transcribed, the template, it's the non-coding, you solve half of the question. Second, if you know that the mRNA sequence is complementary to the transcribed, instead of T is replaced by U, or is the same as the non-transcribed where T is replaced by U. Genetic code table is used to identify the presence of the amino acid, the sequence of the amino acid, for what value or how can we store this sequence. Okay, sequence of amino acid, it depends mainly on the genetic code table. So without genetic code table, we can determine the sequence of the amino acid. Types of mutations. Here in this session, I'm going to introduce them quickly. Types of mutations. We have 3 types: insertion, deletion, and substitution. What the meaning of insertion, what the meaning of deletion, and what the meaning of substitution. First step, first is called insertion. Insertion, what is the meaning of this? Insertion. For example, I inserted here in this part, I inserted a, let's say, nucleotide T. Insertion is the addition form. So here we have AAG is the first nucleotide, first codon, sorry. CTC, second codon. GAA is the third, and TTC is the fourth. Okay, so we have 4 codons. 1, this is the second, this is the third, and this is the fourth. Okay, I'm going to work only with this strand. Okay, not going to work. 3' and 5', we're going to talk about them later. So first we have AAG. So it's going to be A, and the G. I inserted T here between G and C, so it's going to be T. And I have to continue. CT, T, CT. This is the second codon. So here I finished A, G. I put T, T, C, T. And then I have to continue. C, G, A. C, G, A. Then I have to continue. A, T, C. A, T, T, sorry. A, T, T. And finally you have the last one is a nucleotide. Do you see here what we do? We did a shifting here. We did a shifting from the 4th nucleotide. So it was AAG, it's the same, the first nucleotide. Starting from the 4th, we added T, we inserted T, so it became TCT. So here it became TCT. So we shifted by 1. Then CGA, then AAT, then C removed all of them. So this is called insertion. So we added 1 nucleotide. So there's insertion of 1 nucleotide. It's like addition of 1 nucleotide. Okay? This will change in the amino acid sequence. Maybe yes, maybe no. But here, maybe it will, maybe it will not. We will discover this in the coming session next week. If these mutations or these changes will change the sequence of the amino acid. Okay? So this is the first type of mutation. It's called insertion. Let's move to the second type of mutation. Second type of mutation is called the deletion. Deletion, what do we do? Deletion, I deleted 1 of the nucleotide. For example, I'm going to delete the first nucleotide, this one. I delete this one. Okay? Deleting, so they are going to be shifted to this side. So I'm going to start. The first codon is going to be AAGC. AGC. Then we have TCG. TCG. And then we have AAT. AAT. And finally we have TC. Why do we have only in the last one? We have only 2 since this one is deleted. So we deleted 1 here. So this is called deletion mutation. Last type of mutation is called substitution. Substitution mutation. What do we do in the substitution? Substitution, as its name indicates, we substitute. For example, I'm going to substitute the G with A. Okay, it's called substitution. So our first codon is going to be AAA. The first codon is going to be AAA. And the rest is going to be the same. Why? I didn't change anything, so it's going to be CTC. The second codon is going to be CTC. The third codon is going to be GAA. GA and A. And the last codon is going to be this codon, TTC. This is TTC. As you see here, in the substitution, only the nucleotide which is substituted undergoes substitution. Okay? It was G, so it becomes A. While the rest, they remain the same, CTC, GAA, TTC. So this mutation It occurs in a specific point. So we call it point mutation. Okay? So how many types of mutations do we have? We have 3 different types of mutation. First type is called insertion. We added a nucleotide. Second type is called deletion. We deleted a nucleotide. And third type is called substitution, where we substituted 1 nucleotide or more. So it's called point mutation. So it occurs at a specific point. Where insertion and deletion, we will call them later. They are called mutation by frame shift. So we shift the sequence. As in the deletion, we shifted the sequence, or in the insertion, we shifted the sequence. The sequence moves toward the side of the mutation. Okay, guys? So this is everything related to this session, and this is everything to mutation. This is a small introduction for mutation. Next week, we will work more about mutations, about the sequences, and what would happen, and the different types of mutations. Okay? Thank you.